who is also one of the co-organizer of this meeting. So it's a great pleasure to introduce Dr. Alex Garanto. As you know, is the co-organizer of this meeting and is also one of the leaders of the working group 2 in our DARTER action. So Alex is currently an assistant professor at the Department of Pediatrics and Human Genetics at the Radboud University in Nijmegen in the Netherlands. He obtained his PhD from the University of Barcelona in 2011. And after a short postdoc at the University of Barcelona, he moved to the Netherlands to start working with Professor Rob Collin on the design and the characterization of antisense oligonucleotides as a potential approach to treat inherited retinal disease. So Alex is now a group leader and his research focuses on the development of molecular therapies, mainly using RNA-based approaches for inherited neurometabolic diseases and inherited retinal diseases, as well as the development of human-based models to assess them. Alex, we all look forward to your presentation. So the screen is yours.
Yes. So do you see my presentation?
Yes, perfect.
Perfect. So thank you, Aurelie, for the nice introduction. And we have heard during today already about readouts, models, now about genetics. And now it's time to move towards the therapy part. And the aim of this talk is more to talk about, show a little bit of an overview of how therapies are designed and how we can deliver them. And I will not go into clear examples because we have plenty of them afterwards by very nice speakers. Therefore, it's mainly an educational lecture on what we can do. So let me see. So inherited retinal diseases, as it has been already mentioned before, are rare diseases. And we heard, and also Suzanne highlighted now, that it's highly heterogeneous. We have many genes involved in many subtypes of diseases, and still we have several, a portion of those patients that are not yet diagnosed, so they don't know what mutation they carry, which gene is affected, and therefore they are not eligible for a potential personalized treatment that could be developed in the future. But more importantly for IRDs is that there's a progressive loss of vision. That means that we have a window of opportunity where we can act. And the eye, in fact, is at the forefront of therapeutic development. And this is because, well, it's accessible. It's also contained. Whatever we deliver to the eye, in principle, stays into the eye. It's formed by non-dividing cells, so all the photoreceptor cells and bipolar cells, etc., are neurons. And this is something positive because that means that our therapeutic molecule, once injected, will not dilute, but at the same time, it's a con because once those cells are gone, we cannot treat anything. So we need to make sure that we act in time. Also, the eye is an immunoprivileged organ. And as I mentioned, there's a window of opportunity. In addition, if we reach a clinical trial, the readouts are non-invasive, as it was already highlighted in block 1. For example, electroretinograms are not invasive. And also, we all have 2 eyes. So one eye can be treated and the other eye can be the control. But for that, first, we need to know what is the genetic cause, and we need to understand the mechanism of disease. And here, this is an overview on the different techniques, and I will talk a little bit more afterwards, but the sooner we know the genetic cause and the less retinal disease, the progression of the disease is less advanced, more opportunities we have. And in that sense, we can apply what we call genetic therapies, or I like to call them molecular therapies. In more advanced stages, if we don't know the case of the cause of the disease, or the disease is already moderately advanced, we can try to use cell therapy or optogenetics. And eventually, if we don't know the cause of the disease and the retina is quite damaged, we can use retinal prosthetic implants. So what is important when we want to design a therapeutic approach is that we think which type of mutation we want to target. Is exonic, intronic, splice sites affecting splicing. The effect that this mutation has the pattern of inheritance, and of course, the function of the gene. Sometimes we think, oh, we know the mutation, but if we don't know the function, we may not have a readout. We also don't know whether a gene is tightly regulated. It has been shown in other works that if, for example, a gene is delivered and the expression is quite high, actually it's also toxic despite that the mutant one was not producing the correct product. And when we adding the correct one, it was too much. So all these kind of things are needed to know. Also the structure of the protein, the domains, whether it has other functions. And in this case, the organ that we want to target, but in our case is local delivery into the eye, and whether we have an animal or cellular model where we can test that. Also the window of opportunity, as I mentioned, if it's whether we are still in time to treat something or not, and which type of delivery and molecule we want to use. And when we talk about molecular therapies, they were already mentioned in the previous slide, but we can think about gene replacement or gene augmentation. We can also knock out defective genes, and we can regulate splicing with antisense oligonucleotides. We can also degrade the RNA or gene silencing. We can do RNA and RNA editing, optogenetics, and to some extent, within this group of molecular therapies, we can include post-translational read-through, chaperones, or maybe autologous transplantation of cells that have been corrected outside the body. So Luxturna is the most famous genetic therapy in the eye field, and this is because, well, it was the first gene therapy approved in the US. It was the second approved in Europe, and it's also the first gene therapy with market approval to treat a retinal disease. And Luxturna is an AAV, and I will explain later what AAVs are, based therapy to deliver the correct copy of the RPE65, which is a protein involved in the visual cycle. However, despite all the attention that Luxturna had, if we talk about molecular therapies, in general, Luxturna was not the first. And of course, these are non-IRDs, and they, one second now. So they are not IRDs, and but the first antisense oligonucleotide approved by the FDA was actually in 1998 and was vitravin, and it was used to treat CMV retinitis. However, in an age patient, but the problem was that with other treatments that were developed to treat AIDS, though, well, the cases of CMV retinitis were decreasing, and therefore at some point they withdrew the market approval at Europe and the US, and nowadays is discontinued. And it was delivered directly to the eye. The other one, it's actually Macugene, which is aimed to treat neovascular age-related macular degeneration. Again, it's not an inherited retinal disease, but it also affects the retina. And in this case, the FDA and EMA approval were obtained in mid-2000s, and currently is not used in most of the countries because other antibody-based therapies, anti-VEGF, are used. So today, in the next blocks, we have several topics to discuss. In this case, well, gene replacement and delivery, and there will be talks about DMD, LRID3, CNGA, and CNGB. Also antisense oligonucleotide technology, ASHR2A, NR2E3, and ABCA4. And we will finally close with delivery vectors, talking about gold nanoparticles and how they can be used for the retina. And in order to give a little bit of background to what it will come later, so a recapitulation on what gene replacement is or gene augmentation. So if we have a cell with a mutated gene, we can deliver the correct copy of the gene and eventually correct that this gene does the function that it should do, and the cell then will be alive and do the correct function as well. And to deliver the copy of the gene, of course, we can use either viral vectors or non-viral vectors. And this is crucial to know, well, there are pros and cons for all of them. But of course, in the viral vectors, we have integrating vectors that I put here lentivirus and retroviruses. They have large capacities, capacity, but they integrate in the genome, and therefore they can insert the cargo in a place that we don't want. Also, we have the non-integrating, and I already mentioned the adeno-associated viruses, or AAVs, that are the most frequently used, but the cargo capacity is rather small. In the non-viral vectors, we have the particle base with nanoparticles and liposomes in the lead. And also we have naked particles that can be delivered either as DNA, RNA, or oligonucleotides. So for the adeno-associated viruses, briefly, this is the genome of an AAV. It has two ITRs, and then there's the replication and the capsid gene, and other regulatory elements. And basically, we can modify them by replacing whatever it's in between the two ITRs by the copy of the cDNA of the gene of interest, but only the coding region. However, as I mentioned before, 4.7 kb is the maximum that we can include. And that means that most of the mutated genes in IRDs are bigger than that, and therefore they don't fit into an IRD. And the way to produce the virus is because the rep and the cap genes can be delivered in a plasmid without the ITRs in a way that they can in trans express all these genes and create the envelope and pack the virus with the cDNA of interest in the cargo, and that this virus will be able to infect, but it will not be able to replicate because it does not have the rep gene. So in this case, once we have our gene, what is important is how we want to regulate the expression. I already mentioned before that the expression, if it's not well regulated, can be toxic, but also it can be that we don't reach the threshold, the therapeutic threshold. And for that, we can play with the promoter, and we can use a general or constitutive promoter, like the CNV promoter or the chicken beta actin promoter. And that means that the gene will be expressed in all the cells where the virus enter. Or we can use a more specific promoter, like the one of the rhodopsin to get expression in the rods, or RP65 to get expression in the RPE, or whatever promoter cell specific. And in that way, we can control the expression and regulate the cells that we want to target and the toxicity. So those AAVs, and this is a very small list, and nowadays there are many more, 7M8 and ANC80. So all of them are have the produced with the package, the replication of the AAV2. But what it changes is the capsid, and the capsid determines actually which cells or which tissues they can target. And for some of the viruses, we know the AAVs that are, well, the linker or the receptor that allows them to bind and enter into the cell, and for others are not known. And here there are a couple of examples. And how these AAVs work, so they bind to this receptor, they enter into the cell, and they are eventually transported to the nucleus, where they really release the cargo, and it's expressed, and therefore it's secreted to the cytoplasm, where it will be translated to a protein that can do the normal function. And in that way, we are able to produce a protein that was defective in the cell of interest. So now changing topics to the other one that will be part of the talks of today are the oligonucleotides. And basically the talks of today will be about antisense oligonucleotides, or in short AONs or ASOs. And these are relatively small, around 20 nucleotides, DNA, RNA, or synthetic nucleic acid molecules that bind complementary to the pre-mRNA of interest, and they can modulate the splicing, promote transcript degradation, modulate gene expression, or block translation. And actually, this year, within our action, we published this paper on delivery of antisense oligonucleotides, which is a problem in the field, but not for the eye. And here you can see all the different actions that these antisense oligonucleotides can do. And as I said, for systemic delivery, that's still a problem because those antisense go to the liver and can create also immunoresponses or toxicity. But in the eye, they can be delivered as a naked molecules without the necessity of any nanoparticle, liposome, or other mechanism. So directly can be injected into the eye. So if you want to know more, make sure you read this very interesting review. So now we have talked about the molecules, but comes the other question that usually we discuss, and it's how to deliver to the retina. And of course, there are multiple ways to deliver. All of them have pros and cons, and it will depend on the molecule that you use. And here I depict a couple of them. So the topical one, it's eventually the easiest one because it can be just drops on top of the cornea, and the cornea is the outside part of the eye. And this can be really useful if we aim to treat something at the frontal part of the eye. But if we aim to reach the retina, for instance, with an antisense oligonucleotide or with a virus, those molecules need to cross the entire eye until they reach the retina that is at the back. Therefore, it's highly inefficient for that, and also because there are several protective barriers in between. The other option that is the most common one is the intravitreal injection or intraocular. It's more or less easy to administer. All the retinal layers can be targeted. We also cover almost the entire retina, and it's ideal to deliver small molecules. For larger molecules like viruses, at the beginning, they were only transducing the ganglion cells because, as it was explained already before, they are closer to this, the vitreous humor, and therefore they were easy to transduce, but reaching the photoreceptors that are completely at the back part was more difficult. However, nowadays, there are new AAVs that have been engineered in a way that they are able to transduce all the cells of the retina when delivered intraocularly or intravitreally. The other one is the subretinal, and this is the one used at least for Luxturna. And it's ideal to target photoreceptor cells because the injection is done between the photoreceptor cells and the RPE. It can be applied in a specific region, so the ophthalmologist can check where there's a photoreceptors left and inject in there. And in principle, should be suitable for all the molecules because the restriction of the size is not a problem. And if you are not well trained, well, it's technically more challenging, and only small areas can be targeted compared to the intravitreal, and can also cause retinal detachment because of the bubble that is created when the injection is done. And of course, we can try to do systemic delivery, but it happens like with the brain that the retina has its own blood-retinal barrier. So whatever we deliver intravitreally may not reach the retina because it's not able to cross that barrier. That means that also because most of the molecule will go to the liver and the kidney and will be cleared out, eventually high doses are needed, and this can also cause toxicity in other organs. Although the administration is also easy. And the last delivery method is the suprachoroidal injection, which to me is quite challenging, and at least in a mouse, I will not be able to do it. It's and it consists in injection in the suprachoroid space, and based on literature, it diffuses really well and targets the photoreceptor cells, the RP, and also it can target large areas. The only thing is that there are off-target tissues. So all what I found in literature is that the cornea or the sclera also get transduced, and that can be something that we eventually do not want if we cannot regulate really well the expression of the gene. So just to recapitulate in that part, the AAVs, there are 43 ongoing or completed clinical trials according to literature. And for AONs, I checked clinicaltrials.gov, and I could find that there are 6 ongoing or completed clinical trials for IRDs and 2 for other retinal diseases that are not considered IRDs, like age-related macular degeneration. And if we look at antisense oligonucleotides, well, we have, there's 7 genes that have been already targeted with with antisense oligonucleotides, either at the clinical level or already at clinical level. Today, we will hear more about ASHF2A, ABCA4, and NRA2E3, but SEP290 was the first one that was reached the clinic, and it's currently in a phase 2/3 clinical trial. Julio already mentioned it, and I will, in view of time, I will go really quickly. So Leber congenital amaurosis, it's inherited eye disease that appears early in life, and SEP290 is one of the most common, most frequently mutated genes, and in particular one mutation that is the intronic mutation that it's recognized by the spliceosome, and therefore a pseudoexon is included. Initially, it was believed that it was in a 50-50 ratio because in fibroblasts of the patients, it was what we saw, but in the group of Michael Cheatham and Julio already indicated also this morning that using organoids, they were able to see that this ratio actually was 90-10%, and explaining why in the retina it had such an effect. And designing antisense oligonucleotides, this splicing defect could be corrected. And well, since this is just the history of SEP290, in 2006, the mutation was discovered, and the AONs in 2012. There was some in vitro and in vivo work done in several models, and eventually more safety for the molecule that is currently being administered in a clinical trial led by ProQR. And I think actually Arthur Sidersian is in the call today, so you can talk to him eventually via the chat. And the clinical trial of this molecule showed that when injecting the worst eye, there was actually an improvement based on best corrected visual acuity, and it was also maintained in time based on the results of the interim results of this phase 1 clinical trial in this case. And with those results, they were able to start a phase 3, where now the injections, instead of being done every 3 months, are done every 6 months. And yeah, what this showed is that the AONs are well tolerated by the human retina, and they are promising tools for therapeutics. And with this, I would like to finish and just mention that the eye is at the forefront of therapeutic development, that local delivery to the eye is very efficient and safe, that AAV-mediated therapy is being studied for several subtypes of IRDs, and antisense oligonucleotides are well tolerated by the eye, and several AONs are also being assessed at clinical trials. And of course, there are many other molecular therapeutic strategies that are being developed, like genome editing, chaperones, optogenetics, but in view of time, I don't have time to talk about all of them. I will thank to all the group, and of course, all of you for being here and listening. And if you have any questions, I'm happy to take them.
Thank you very much, Alex. It was a great presentation, and I think it was a great overview of all the tools that we will hear about this afternoon. Again, you can write your questions in the chat. We have a few minutes, and waiting for the questions, I can start. So I have several, so I just have to limit myself. You mentioned the other type of therapies at the end, like the CRISPR approach, and I just wanted to have your opinion, because it's something that I get asked as well, like working with oligos at the moment, people, and considering the progress with the genome editing approaches, especially with the prime editing, and the less and less off-target effects, people are asking, do you believe, how do you see the future of AON therapeutics in a way, or classical gene therapy compared to genome editing approach?
I think for genome editing, we are still, for certain types of mutations, we are still far away. I mean, you need to have, for example, what in the eye, what it's done is for the same mutation of SEP290, they are removing the intron. This eventually it's easier because you just cut and remove. And the one that was done by stock therapeutics systemically, it's more about degrading a protein that is not a mutated gene to avoid the dominant effect, that kind of. So I think for those, there's more promising, but I think there's still a lot to do that with AONs or antisense technology, we can still, we have more data and we can still target them easily than with genome editing, because I think the off targets also genome editing are a little bit, we should be more careful, because the AON eventually will dilute. So you can stop your treatment when with CRISPR, you modify the genome, and you need to make sure that you are not changing anything else.
Yeah.
So, and I think we need to work more on developing this genetic safety and how these molecules can be more safe when delivered.
Okay. So there's one question from Jérôme Roger in the chat. How much progress have been done to identify AAV targeting photoreceptors through intravitreal injections?
Well, there are several reports in literature, several of them by the group of Luc van der Berge, where they have investigated several AAVs and do comparisons in mouse, in monkey, and how they transduce the retina. And I think in that way is how they found several serotypes that can really target the entire retina when delivered intravitreally. Otherwise, the AAV8 and 9 usually are the ones that most people use for targeting photoreceptors. But intravitreally, the 7 and 8 seems to work quite well. And there are a couple of other ones. I can write the name of them.
If you have the reference, I'm interested as well. It's a very good question.